Hi. So, in our last discussion, we looked at how speciation happens primarily through the mode of reproductive isolation. Now, if you remember our discussion right in the first week, we said that Darwin said that the entire diversity that we see around us, that has arisen because there has been continuous rounds of speciation over the last billions of years, because of which through descent with modification over time, starting from some simple ancestors, the entire biodiversity around us has arisen. Now, if you think about it very closely, there is a bit of a missing link here. What is that thing? So, when we were discussing about speciation, we saw in what way reproductive isolation sets in, such that two populations which earlier were able to exchange genes, now they are not able to exchange genes anymore, and thus they have become new species. But when they are becoming new species like that, they are essentially becoming new species of the same type. In other words, when two salamander species or two bird species, they are becoming reproductively isolated from each other, they are becoming two different species of salamander, they are becoming two different species of birds. They are not really converting it into something very different. They aren't converting into, let us say, a snake or a mammal or something like that, right? In other words, for the mechanisms of speciation that we talked about, those are talking about how more groups of the same type can arise. But if we have to give credence to Darwin's original claim that descent with modification has led to the entire biodiversity, then we have to have a way to figure out how exactly do biological organisms lead to new traits, okay, or rather generate new traits. Until and unless you explain how biological novelty originates, it is very hard to see how you can go from simple organisms to more and more complicated organisms, or things like the peacock's tail evolving, etc. All that one cannot even think about unless one understands how biological novelty originates. And that is something that we are going to talk about in today's discussion. As I am telling you over here, this treatment is primarily after Fukuyama and Kirkpatrick, but there are one or two extra things here and there that I might be able to say. Now, this particular question, where does novelty come from? Of course, Darwin thought about it, many other people thought about it, and one of the things that they realized is that in many cases, what shows up as a trait today is essentially some repurposed organ of some that arose for some other purpose. So, for example, think about us humans. We have these opposable thumbs, right? And because of that, we are able to grip things, and this is supposed to be very important in the evolution of humans, because this ability to grip is what allowed us to be able to make tools and use tools. Now, turns out we are not the only organisms who are able to grip things. Another famous example of things which can grip are the giant pandas, and you can see how it is able to grip this bamboo and eat that. But if you look at the panda's thumb and compare it with the human thumb, you will find a crucial difference. What is it? So, in us humans, the thumb, which is this one, is the fifth digit in the hand. Now, in the context of the pandas, if you look at it, they will have all their 5 digits. So, the fifth digit is the one that is homologous to our thumb, but the fifth digit is not the opposable one. The opposable one is a 6th digit, okay, over here, and that is not really a digit, that is essentially one of the wrist bones which has become modified to take that purpose. In other words, in many cases when organisms are trying to evolve a new trait, all that they do is they take an existing thing and somehow modify it, change it in some way such that it can be used in a new way. Now, this repurposing is not only something that happens at the level of the organism, it can also happen at the levels of genes. So, I will give you one very nice example. So, for example, what I am showing you is a schematic of the vertebrate eye, and you know that in the vertebrate eye we have this lens over here, and this lens is very important because it gathers the light and then focuses it on the retina, from which through nerve conduction the signal goes to our brains. Now, obviously, in order for the lens to be able to do this, it has to be a transparent thing. If it is not transparent, light will not pass through it. So, in vertebrate lenses, the main protein that takes care of with this function, this is the transparent set of proteins known as the crystallins. Now, turns out that different vertebrates have different combinations of crystalline proteins in their lenses. So, all of them, the eye is doing the same job, okay, the lens is doing the same job of letting the light pass, but the proteins that are combining to form those lenses are different. And just to show you how different they are, so here are a number of proteins, crystalline proteins in various kinds of organisms, going all the way from frogs to humans to rabbits. But what is more important than this is this list over here. So, these are the list of the ancestral proteins and their functions, which has finally led to the clear crystalline proteins in the eyes. So, here you find that a large fraction of them are actually heat shock proteins. So, these are proteins which are supposed to be stress resisting proteins, and as the name suggests, typically related to heat shock, withstanding heat shock, but also playing other kinds of roles. Then there are other, for example, this is a reductase, NADPH dependent reductase. Then you have other kinds of enzymes, this is the dehydrogenase, there is a deaminase, and so on and so forth. I mean, one of them is alcohol dehydrogenase gene, the one, for example, which forms the crystalline protein in the camels, right? This one over here, zeta. So, this tells you that the crystalline protein has actually arisen in different organisms at different points of time, which is what is shown by that phylogeny, and in each case each lineage has actually taken a very different protein and has ended up repurposing that for the purpose of forming the lens, right. So, this tells us that proteins do change their function and they can change their function. Now, there are two ways in which a protein can change its function. What are the two ways? First is that it can change its structure itself, the amino acid structure, which obviously requires it to change the sequence of the protein coding gene itself. And the second thing is, instead of changing the structure, it can also change its expression pattern, how much is being expressed, where is it being expressed, and so on. Of course, a third thing is that there can also be post-translational modifications that can also lead to change in its function. Now, if a protein ends up changing its structure or its expression pattern, then what is going to happen to its original function? Because after all, if the protein is there, it has to be playing a role, right? So, if suddenly a heat shock protein, a stress resistant protein starts forming the eye lens, what is going to happen to the heat shock function? So, there has to be a way by which the original function needs to be retained in the organism, and then on top of that the organism, the protein should be able to take a new function. So, how exactly can this happen? Now, turns out that there are many ways in which this can happen, and the first and the simplest way is to form a new gene. So, where exactly, how exactly will a new gene be formed? Where will it come from? So, turns out that there are four major ways of gene formation, and these four major ways is what we are going to cover in our discussion today. And these are, as the list says, gene duplication, exon shuffling, de novo gene birth, and horizontal gene transfer. So, we will start with gene duplication followed by mutations. So, what exactly is gene duplication? So, as the name suggests, duplication of an existing gene or a large chromosomal segment containing a gene. So, two or more genes that have arisen due to duplication, these are known as paralogs. Whereas, if you have one gene which over time due to mutations or other things has mutated and a new form of the gene is there. So, these two are related by directly by ancestry, these are known as orthologs. So, what is the logic of gene duplication? How does it solve the problem? So, the whole idea is that if you have multiple copies of a gene, then one copy will keep on performing the original function, whereas the other copy, now it is in some sense freed up, and now it can mutate and it can now take other functions and so on. So, turns out that gene duplication plays a very key role in evolution, and about 1% of human genes have been duplicated every million years. Now, think about it, that is although million years sounds very big for us, evolutionarily speaking it is just a blink. So, 1% of human genes have been duplicated every million year, and about 1400 duplications have been fixed in humans and chimpanzees after we diverged about 7 million years ago. So, this tells you how critical this thing is. Now, what are the ways in which genes can be duplicated? So, there are 3 or 4 ways in which it can happen. The simplest way is what is known as unequal crossing over. So, when you have meiosis, during that time the gene segments are supposed to be exchanged, but instead of exchanging, what happens is that in one chromosome there is a duplication, and the other chromosome this thing does not come, and it just there is a deletion over there. So, in this way, if the part that is getting duplicated contains a gene, then there is a gene duplication that is happening on one of the chromosomes. The other way in which it can happen is what is known as replication slippage. So, during DNA replication, sometimes the DNA polymerase it gets dislodged from the DNA, and then it sort of comes back, but in the process of doing that, sometimes it ends up copying the same sequence more than once, and this is particularly common in those regions of the genome where there are repetitive sequences. The third way in which it can happen is retrotransposition. So, suppose you have a gene and the gene has transcribed into a mRNA, but then after that, instead of going into the translation process, the transcribed mRNA is reverse transcribed into a complementary DNA or a cDNA, and then this cDNA it comes back and gets reintegrated into the genome at some random location. So, this is what is known as retrotransposition, and obviously this can lead to the formation of new genes. Now, note over here, since this thing is happening from the mRNA, the introns are cut out in the mRNA, right. So, the cDNA typically does not have the introns, and therefore, a simple way by which people can recognize that a gene has arisen due to retrotransposition of a different gene, obviously there will be sequence homology, but at the same time the intron portions will be missing in the new gene. So, that is how you know it is a retrotransposed gene. And of course, the other way in which you can have gene duplication is if the entire genome ends up duplicating itself. So, when it is happening within the same species, that is what is known as autopolyploidy, and as we have discussed, this is something very common in plants. Now, let us look at some examples of gene duplication. So, this is a very beautiful looking langur. This is the red-shanked duke. It is found in Southeast Asia, and turns out that unlike most or unlike many primates, this one actually its leaves. Okay. Now, you know that leaves they contain cellulose, and therefore, leaves are very hard to digest. So, the other group of organisms which can digest leaves are the herbivores, and many of them they have these 4-chambered stomachs, and they have bacteria, symbiotic bacteria which live inside the gut, which helps them in the digestion. So, essentially what happens is that these bacteria they end up digesting the leaves, and then the organism, the host, ends up digesting the bacteria, and that is how it gets the nutrition. Now, turns out that in this particular duke langur, they use one of the enzymes known as the RNase 1B. And this RNase 1B digestive enzyme, it rose by gene duplication about 4 million years ago, and the gene from which it arose is the pancreatic ribonuclease gene RNase 1. So, the RNase 1 gene is still there inside the Duke, but the RNase 1 gene, it's actually, its function is entirely non-digestive. So, after the RNase 1b gene was created, then it actually acquired lots of mutations very fast. So, there were 9 amino acids that ended up changing, and because of these things the RNase 1b became a digestive enzyme, and more critically now it was able to function in the small intestine where it had, it could function in a slightly more acidic environment. So, these mutations, this enabled this thing to function in acidic environment, which was really good for these guys, the Dukes, to be able to become leaf eaters. So, this is how a new function is being gained. Now, another very important example of gene duplication is in terms of the vertebrate sodium channel. So, you know that in our nerves we have these voltage gated sodium channels, and these are very important for the purpose of nerve firing. Now, if you look at the structure of these genes, you find that these are composed of 4 domains, and each domain in turn is composed of 6 units, 6 transmembrane segments. Now, if you look at the sequences of these transmembrane domains, these transmembrane proteins or segments in each domain, you find that the 6 in domain 1, you can call them 1, 2, 3, 4, 5, 6, the sequences are extremely similar to the 6 in the second domain, the third domain and the fourth domain, which basically means that the first segment of domain 1 has the same sequence as the first segment of domain 2, as the first segment of domain 3, and so on and so forth, and so on for all the 6 segments. So, this suggests that there is probably some relationship over here, and people actually figured out that this entire thing happened because there was a duplication, or rather 2 duplications, one after the other. So, first one became 2, and then each one of them gave rise to 2 more. That is how you ended up getting 4. And so, from one locus you ended up getting this entire thing. Now, this was the example of gene duplication, and now we are going to talk about the special case of gene duplication, which is retrotransposition. So, in human beings we have this gene known as the phosphoglycerate kinase 1, PGK1, and this is a very important gene because it is involved in the glycolysis. So, it is found on the X chromosome, and it contains multiple introns, and it is expressed in all over the body. So, it is a very important gene. Now, turns out that human beings also have another gene called the PGK2 gene, and this is found in the 6th chromosome. And very interestingly, when you look at the function of the PGK1, the sequence of the PGK1 and the PGK2 genes, you will find that there is a huge amount of similarity between them, except for the fact that the PGK2 gene does not have any of the introns that are present in PGK1 gene. So, this clearly tells you that this gene has adhesion, PGK2 has adhesion due to retrotransposition from PGK1, which has moved it from the X chromosome to the 6th chromosome. And turns out that this PGK2, now it is under the control of a different promoter. Now, this is very important. See, retrotransposition is simply going to take the cDNA and it is going to put it somewhere else, randomly somewhere else in some other place, or sometimes in the same chromosome, does not matter. But wherever it is going, it has to be reasonably close to a promoter region or within the effect of a promoter region, without which the gene is not going to be expressed. So, this is the problem with retrotransposition. Until and unless it gets integrated in a opportune place where it is under the control of different controlling elements, without that the gene is not going to work. And in this particular case, turns out that the PGK2 gene did find itself in the vicinity of a proper promoter, and now today this gene only expresses in the testes of humans, and because of that it actually ends up playing a big role in sperm motility in humans. Now, Fine. So, we saw how genes duplicate, we saw examples of genes duplicating. Now, the question is what exactly happens when a gene is duplicated? Now, there are two major possibilities. One possibility is that, as I said, the duplicate gene is not even functional to begin with. This can happen in many ways. It can happen because suppose the position, the part that got duplicated does not contain the entire gene. So, it is just a fragment, does not work, or as I said, particularly in the case of transpositions, it does not have the regulatory elements that are needed to express the gene properly, in which case it will not function, or in many cases the post-translational transcriptional splicing regulation is very, very important. But suppose the duplicate has been formed due to transposition, in which case the introns are not there, which means that all the post-transcriptional splicing regulation will not happen, which in turn might mean that the gene may not function. So, in all these cases, we are going to get a duplication event, we are going to get some fragment either full or incomplete, but that fragment will not lead to any product. And in these cases, such non-functional genes, they are known as pseudogenes. The point with the pseudogene is, because it is non-functional, it is essentially selectively neutral, which means that it will end up gathering mutations in the genome and it will keep on becoming more and more unlike what it rose from. So, basically selection will not work on this. On the other hand, the second possibility is that you actually have a functional gene. But if you have a functional gene, then there are 4 possible fates of a duplicated functional gene. What are those? So, the first one is that suppose the gene is functional, but it is not really providing any fitness benefit. Okay. In other words, whatever the original gene was producing, the quantum of that is sufficient, and therefore, this new gene, the organism does not even feel whether it is there or not there. Now, if that happens, then there is a possibility that this might get lost, because any bad mutation, say deletion or some kind of other kind of things that lead to loss of function mutation, none of those are going to be seen by selection. Okay. And therefore, this at some point there is a possibility that this might become an inactive gene, in which case it will again become a pseudogene. The second possibility is that the duplicates actually retain their original function, and this retention is something that is selectively favored. When can this happen? This can happen if the gene product, the amount of the gene product that is needed is somewhat large, and having an extra copy of the gene is allowing for extra amount of protein in the body, that particular protein whatever is coded by that gene, and that is beneficial for the organism. So, if such a thing is happening where increased quantity of the protein is useful, then there is another possibility that there might be subsequent duplication events, because of which the number of repeats in the genome actually, or number of duplicate genes for that particular original gene keeps on increasing, and it actually varies between individuals in the population. So, this can lead to variation in the number of copies of the gene that various individuals in the population carry, and this phenomena is what is known as copy number variation, okay, or CNVs. And as I said, CNVs can affect the gene dosage. So, that the level of the product of the gene in the body of the organism, and in that sense it can become a very significant source of genetic variation. Now, this was number 2, CNV. The third thing that can happen is what is known as new functionalization, which is that the redundant gene copy can now start accumulating mutations, and in that process it might acquire a new function, thereby leading to some kind of a novelty. And we have already looked at one such example, the RNase 1b in the red-shanked Duke langurs. We will look at 1 or 2 more examples as we go along. And the fourth thing that can happen is known as subfunctionalization. What is that? Now, suppose the original gene was doing more than one function, okay, and when there are 2 copies, now one copy takes care of one part of the original function, other copy takes care of another part of the original function, such that what was the job that was earlier done by one generalist gene, now there are two specialist genes doing two parts of the job. Okay. So, this is what is known as subfunctionalization. So, I will give you examples of all these things: copy number variation, neofunctionalization and subfunctionalization. Now, sometimes, not always, the two can even happen hand in hand, neofunctionalization and subfunctionalization, and I am going to talk about that too. So, let us talk about an example of the copy number variation. Now, you know that we humans, we have this enzyme called alpha amylase in our saliva, and this is very important because this allows us to break down starch. So, starch digestion starts right in the mouth, which is why if you take some bread and you chew it for slightly longer, the entire thing starts tasting sweet. The starch has started breaking down into its component, glucose and other things. Now, this particular enzyme alpha amylase is coded by a gene called AMY1, which shows copy number variation in humans. Now, it is known that if you have more copies of AMY1, then you have more alpha amylase in your saliva, and if you have more alpha amylase, then that means you are going to become better in starch digestion. Now, this line of reasoning is leading to a prediction. What is the prediction? The prediction is that different populations have different levels of starch that they eat. Some people or some populations, individuals eat a lot of meat, let us say, and there are other places where individuals eat lots of starch and everything in between. So, the prediction over here is that populations whose diet are more meat based, they do not really need the alpha amylase thing. So, there the number of copies of alpha amylase will be, sorry, it should be the other way around. Populations whose diet are more meat based should have lesser number of copies of AMY1. Sorry, whatever is written in the slide is wrong, just correct that. So, populations whose diet are more meat based should have less number of copies of AMY1 compared to those whose diet have more starch. So, do we have any evidence to believe this? Turns out that we do. So, these people, Perry et al., what they did was they looked at different populations, where the amount of meat that they eat versus the amount of starch that they eat are very different. So, here I am showing you, example from one individual each from the Biaka tribe of Africa. So, these are hunter gatherers and their diet is highly meat based. They are being compared with an individual who is a Japanese, from the Japanese society, which as you know is a more modern society, and there the amount, of course they do eat meat, they do eat fishes, but the amount of starch that they eat is much larger than, for example, compared to the Biaka. So, what Perry et al. Did was they extracted tissue from both the Biaka and the Japanese, and they ended up looking at their chromosomes by coloring them with red and green color. So, each red green thing that you are seeing over here, this is showing you the AMY1 locus in that individual. So, you can simply count the Biaka individual, this has got 3 copies on one chromosome and 3 copies on the other chromosome. So, a total of 6 copies of the AMY gene in this individual. Whereas, if you look at the Japanese individual, on one chromosome this person has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and the other chromosome that person has 4. So, this person is carrying 14 copies of the AMY1 locus. So, this is clearly showing you how there can be huge variation in terms of the copy numbers. However, whatever I showed you over here, this is just one individual from the meat eating population compared to one individual from the starch eating population. But the copy number variation definition that I gave you, that talks about this stuff at the population level. So, obviously, in order to understand this properly, you need to look at the population level. So, turns out that they did the same paper, looked at this problem. So, here what they did was they took 7 populations, 3 of those populations were incorporating diets which had a lot of starch, whereas 4 of the populations had traditional diets which had either little or no starch. So, they call the latter the low starch and the former the high starch diets or high starch populations. And here on x axis they have the AMY1 diploid gene copy number. So, over both chromosomes how many copies does each individual have, a histogram of that, and what proportion of individuals have that many copy numbers, that is what is on the y axis. It is a normal histogram, and this orange thing shows the low starch populations and the blue thing shows the high starch populations. So, you can see that the low starch populations, their proportions are larger in the low copy number case, whereas the moment you go somewhere above 6, typically it is the high starch populations which have greater number of greater copy number, basically telling you that on an average the high starch populations have much greater copy number compared to the low starch populations. And how greater was greater? So, on an average the populations with high starch diet have 6.72 copies of AMY1, while those which have a lower starch diet they have about 5.44 copies. Great. So, this was an example of copy number variation. Now, we will look at an example of neofunctionalization. So, we have already looked at one example, RNase 1b. We will look at even more stark example, that of color vision in some primates. So, in our eyes we have a gene, what is or rather we have multiple genes known as opsin genes, and these lead to the formation of opsin proteins. Now, these opsin proteins, they lead to the formation of pigments, different colored pigments, which have their maximum absorption in certain wavelengths. Now, the way we see color, or way all primates see colors, all vertebrates see colors, is through the action of these pigments. And the more the number of pigments that you have, the more the number of colors that you will be able to see. So, if you look at ancestral primates, they had 2 opsin genes, one was on chromosome 7 on autosomal 1 and the other was X-linked. So, the gene that was on the autosomal on chromosome 7, the autosomal one, the protein coded by that was able to absorb the light at the short wavelength, so the blue side wavelength, whereas the other one which was on the X chromosome, that one coded for a protein which was typically absorbing in the longer wavelength, the reddish side. So, because of this, ancestral primates, their vision was dichromatic, which means that they could only see those colors which could be created by mixing these 2 pigments or the effects of these 2 pigments, and therefore, the color vision in these primates was relatively limited. However, at some point in the ancestors of apes and the old world primates, who are the old world primates? The baboons, the langurs, the macaques, these are the old world primates. So, at some point in their ancestors, there was a gene duplication that happened, and this happened around 35 million years ago, and this happened for the gene that was there on the X chromosome. When that happened, this was a tandem duplication, which means that the new copy was placed right next to the old copy on the X chromosome. Now, when this happened, then one copy, it actually retained its original function, and this is what today is known as the long wavelength opsin, and this is one which absorbs light maximally at 560 nanometer. The second copy actually started gathering mutations, and people have even shown what mutations those are. There are 3 mutations that happened one after the other, and because of these 3 mutations, the optimal wavelength that it could absorb went down to 530 nanometer. Not much, just 30 nanometer, that is it. But now, because of this reduction, now it was able to detect the medium wavelengths, and therefore, suddenly from a dioptic vision, the apes and the old world primates, they were able to now have a trichromatic vision, because of which they were able to see many more colors. And this actually is supposed to have been a major evolutionary boost to these two lineages, the apes and the old world primates, because when you are seeing more colors, obviously you are able to make differentiate the world much better. So, one way in which people have hypothesized that this might have played a huge role is in terms of their ability to distinguish a ripe fruit from an unripe fruit in the background of a foliage, the green background of a foliage. So, the old world primates with their trichromatic vision are supposed to be much better in terms of doing that as opposed to the new world primates, and this is supposed to be a evolutionary advantage that evolved because of neofunctionalization of that gene on the X chromosome. Now, a quick example of subfunctionalization. So, remember, subfunctionalization is where the original gene has a broader function and then there is a gene duplication, and the function of that now becomes original general function now becomes partitioned into two or multiple genes. So, this example comes from the zebrafish, very popular model system Denio-Radio. Now, in zebrafishes, they have a certain gene called the engrailed gene, whose product helps both in the formation of muscles as well as in the formation of the nerves in the retina. Now, turns out that in these fishes, there are two such genes, engrailed genes, so called engrailed ENG1A, ENG1A and ENG1B. And these two genes were formed due to a duplication event in the teleost fishes, the ray-finned fishes in that lineage. Now, turns out that the ancestral capital ENG1, that ENG1 is still found in mammals and birds, and in both these organisms, both this group of organisms, ENG1 is expressed both in the head and in the muscles, right. But when it comes to the zebrafish, ENG1B expresses only in the hindbrain and in the spinal neurons, whereas ENG1A is expressing itself in the pectoral appendage bud. So, basically over here. So, which is suggesting that the earlier function of getting expressed all over the body is what the ancestral gene was doing, and now when the subfunctionalization has happened, one copy of the gene is expressing at one place, the other copy of the gene is expressing at another place. So, this is a spatial subfunctionalization that has ended up happening. Now, turns out that every mechanism that I showed you, all of that, it is not that these are happening in a vacuum, it is not that they are happening one at a time. Sometimes these things actually end up happening together, leading to a very fascinating array of molecules, and one such example that I am going to share with you is that of the globin gene family in primates. Now, you must have heard of hemoglobin, right, the molecule which allows us to carry oxygen and carbon dioxide in our blood. You might have also heard of myoglobin, right, the molecule which stores oxygen in our muscles. Now, turns out that hemoglobin and myoglobin, these two are actually coming from one ancestral gene, and that divergence happened about 600 to 800 million years ago. So, that is not what is shown over here. Now, please note, whatever I am showing you over here, this is not a phylogenetic tree. This is just a flow chart showing how the divergences happen, but I have put the values at some places. So, after this divergence happened between hemoglobin and myoglobin, the protoglobin gene, at some point it underwent a duplication, and then after the duplication there was a lot of divergence that happened due to mutation accumulation, but they were still on the same chromosome. So, at some point you had two gene copies which had become sufficiently diverged. One led to the alpha globin family, the other led to the beta globin family. So, the alpha globin family is this one, and the beta globin family, the ancestor to the beta globin family is this one, this is the ancestor to the alpha globin family. Then at some point there was a transposition that happened. Now, because of that transposition, the progenitor of the alpha globin gene that came on the 16th chromosome, whereas the progenitor of the beta globin that came to the 11th chromosome. Now, where the ancestral one was, I could not figure that out, but what we know today is that one set is on chromosome 16, the other set is on chromosome 11. Now, about 300 million years ago, the progenitor of the alpha globin family that divided into two parts. One part became the so called zeta family, and the other went towards the alpha family. Then about 40-50 million years ago, again there were more duplications and three such genes were formed, alpha 1, alpha 2, and one more gene psi alpha 1, which was formed, but it essentially died after that, it became a pseudogene. Similarly, at this end, the zeta gave rise to what is known as the Z globin gene. It gave rise to another one, but that one also died. So, together all these form what is known as the alpha globin family. On the other side, what happened was about 150 to 200 million years ago, there was three duplication events. So, you have the progenitor of the epsilon, which gave rise to the epsilon globin, then you have the progenitor of the gamma globins, and then at some point about 100 to 140 million years ago, that again divided by duplication to lead to two different things, the G gamma and the A gamma forms. And then the third thing that had arisen, that was the beta ancestor of the beta thing, and that beta ancestor about 100 to 140 years ago again ended up duplicating and diverging and forming three different genes. Today two of those are still alive, two of those genes are still functional. One is called the delta, the other is called the beta, and one of them actually ended up dying, and that is the pseudo psi beta pseudogene, and all these together form the beta globin family. So, you can see that this entire thing has happened due to duplication, divergence and non-functionalization. But this stuff that has happened, the consequences of this are amazing, and now we will discuss the consequences. So, as I said that the hemoglobin and myoglobin, they diverged about 600 to 800 million years ago, and looks like that happened because there was a whole genome duplication event. Now, because of this, there was a very nice physiological division of labor that ended up happening. Why? Because the globin family is able to attach to oxygen. However, the way it attaches, that is going to determine what the function is going to be. So, for example, if you want a molecule to store oxygen, then you need it to bind to it somewhat strongly. So, that is what happened to myoglobin. Whereas, if you want something to act for transport, that is something that should be able to hold it, but not so tightly that it will not be able to give it up. So, it has to hold it in an optimal way such that under certain circumstances it will take the oxygen, certain other circumstances it will give out the oxygen, right, and that is what hemoglobin specialized in. But just one hemoglobin molecule, or rather one globin molecule, would not have been able to do it. So, what happened is that the protohemoglobin gene that duplicated again and giving rise to the progenitors of the alpha and the beta globin genes, as I showed you. This happened about 450 million years ago. And after that, there was a lot of functional divergence due to duplication and other things, because of which finally we have so many alpha family and so many beta family genes. Now, turns out that the hemoglobin that we have today, it is a tetrameric molecule. There are two molecules from the alpha and two molecules from the beta family, and that is why it has a alpha 2 beta 2 configuration, and this configuration of having 4 different globin proteins coming together and forming one big huge protein, this is what has allowed hemoglobin to become so good in terms of transporting oxygen. And this would not have been possible if all these different globin forms had not evolved through all those gene duplication and so on. But this is not all. There is more to the story. What is it? Remember I told you about one form called the gamma globin, right? Now turns out that subsequent duplications within the beta globin gene family resulted in multiple stuffs including the gamma globin gene. Now this gamma globin, these genes or the proteins that they are coding, these proteins are actually part of the fetal hemoglobin protein. Okay. So in mammals, and particularly in primates, the fetal hemoglobin has a very different property compared to the adult hemoglobins. Why is that so? That is because, think about it, the fetal hemoglobin needs to be able to extract oxygen from the mother's blood, right. So, if the fetal hemoglobin has hemoglobin, or if the fetus is carrying hemoglobin that has the same properties as the mother's hemoglobin, then it will not be able to take the oxygen from the mom's blood. That is why the fetal hemoglobin needs to have a greater oxygen binding capacity, which it accomplishes by having 2 alpha units combined with 2 gamma units. Okay. This innovation is what allowed mammals to evolve, because only then the formation of the placenta and the fetus being able to extract oxygen from the mother's blood supply could evolve. But that is not the whole story yet. So, in most vertebrates, the same hemoglobin ends up transporting oxygen throughout their life. But as I showed you that in mammals, it is the fetal hemoglobin which is transporting oxygen during the fetal stage. Now, this is excellent as long as the fetus is inside the mom's body. But at some point the fetus comes out, it is born. When it is born, after that, if the fetal hemoglobin continues, then that is going to be a problem for the independent offspring. Why? Because now the independent offspring has to have its own oxygen supply, right? And at that point, if the fetal hemoglobin has a higher affinity for oxygen, then that is going to be an issue. Therefore, a few months after birth, fetal hemoglobin actually is no longer made, and that is when the entire oxygen transport shifts to the adult hemoglobin, hemoglobin A, which is alpha 2 beta 2. And this shift again is very, very crucial for independent life. So, after the evolution of the gamma subunit through duplication, there has been a temporal subfunctionalization between the beta and the gamma subunits. In other words, earlier the alpha 2 beta 2 HBA was able to take care of the entire thing of the life of the entire oxygen supply throughout the entire life. But now, because of the mammalian way of life, you need the gamma thing, the fetal hemoglobin, but you need the fetal hemoglobin only for one part of the life. For the other part of the life, you need the other one. So, there has been a temporal subfunctionalization between the beta and the gamma subunits in terms of forming the fetal hemoglobin and the adult hemoglobin. And this entire thing was crucial for the evolution of live birth in mammals. If this had not happened, then the evolutionary innovation of live birth, okay, the placenta and the fetus getting all the nutrition from the mom, etc., none of this would have been possible. So, this shows you how all these gene duplication, neofunctionalization, subfunctionalization, how all these things are actually interacting with each other in a rather complicated dance, so to speak, in order to lead to novel functions in biological systems. Now, so we started by asking how does one create new traits, and we talked about formation of new genes, and then we are done with gene duplication. Now, we are going to look at another way of doing the same thing, which is exon shuffling. So, what is exon shuffling? So, exon shuffling is when you know what exons are, right. So, you know that in many organisms, the genes do not come in one continuous ATGC sequence. So, you have I am sorry, I will take that back. They do come in a continuous ATGC sequence, but the final thing that is going to be translated, that thing does not come in one continuous stretch on the genome. So, what happens is you have stretches which form the final, which have information about the final protein that is going to be coated, and these are known as exons, and between the exons you have stretches of DNA which after the mRNA is transcribed are cut out, and these are known as introns. So, two or more exons, the protein coding things from different genes can be brought together ectopically, means from the outside, or the same exon can be duplicated to create a new exon intron structure, and this process is what is known as exon shuffling. So, just to give you a visual feel for this, let us have these two genes, gene 1 which has 3 exons 1, 2 and 3, and gene 2 which has again 3 exons, let us call them A, B and C. So, let us assume that what happens is that this part B and C, this comes and this gets shuffled into this intron between 1, 2 and 3, and this 3 part, this now comes over here. So, what you will get is exon 1 intron, exon 2, and then this intron gets modified because these two exons go and sit there, and the 3 thing that this exon 3 from here actually shifts to this one. So, what I am showing you over here is a symmetric case, but it does not really need to be symmetric. It is entirely possible that let us say B and C from here just come and sit over here, and then you have 3 over here. So, there does not need to be a reciprocal exchange, that is all I am trying to tell you. Now, how does this happen? Retrotransposition is the simplest way in which it can happen. There is something also known as illegitimate recombination, things that should not end up recombining. I will not go into the details, you can figure out the mechanics on your own. And the resulting protein, that ends up being a mosaic of many different domains, and sometimes it is just a hodgepodge, goes nowhere, but sometimes it can actually potentially lead to a novel function. So, I will give you actually I will give you 2 examples of this. So, in humans and in many other organisms, we have something known as the tissue plasminogen activator. Okay. It is a enzyme that breaks down blood clots, and because of that it is very, very important, because if it is not there, then obviously the chances of your getting a stroke or a heart attack is increased several folds, which is why it is medically it is a very, very important enzyme. It is used tremendously for treating strokes all over the world. Now, if you look at the structure of this enzyme, you will see multiple domains. So, there is one domain, this is a protease domain, this is where the cutting actually happens, but there are other domains, for example, these 2 Kringle domains and this EGF domain and this finger domain and so on and so forth. Now, what people have done is they have taken the sequence of each domain separately and they have tried to map it with sequences of other genes for other proteins, and doing so they have actually figured out that the exons of these genes have actually all originated at different places. So, for example, this finger module that I am talking you over here, this has come from a gene known as the fibronectin gene. Okay. Similarly, this protease module over here, this has actually come from the trypsin gene, and this EGF module that I am talking about, this is essentially the epidermal growth factor, something which you are studying about cancer, you will come to know about it, but you can see it is a growth factor thing. So, essentially this entire gene is a hodgepodge of exons from different places all coming together and forming a novel gene with a novel function, which nevertheless is extremely important for the organism. The second example that I am going to give you for exon shuffling is locus called the Jingwei locus. So, this locus is found only in Drosophila teixeiri and Drosophila jacoba, two species of Drosophila, and the way this locus is formed is very interesting. So, this is the Jingwei locus. So, in the Jingwei locus, if you look at it closely, you will realize that the first three exons, these three exons are actually just duplicates of another gene known as the yellow emperor gene, the YMP gene, right. So, these three exons along with the two introns in the middle are these. Now, so this is one duplication already. Now, what has happened is, this is Drosophila, right. So, these live on rotting fruits, etc. Therefore, their ability to tolerate alcohol is a very big thing, because rotting fruits are typically high on alcohol, and therefore, if you can't tolerate alcohol and you are a fly, then you are in trouble. So, in order to break down alcohol, they have these things known as the alcohol dehydrogenase, ADH gene, right. So, what has happened is that after these three exons, there is a large intron in the YMP gene. So, this entire thing, the entire ADH gene minus the exons, but along with the stop codon, this entire thing actually has gone and it has now started sitting over here, right, in the middle of this intron. Now, how can this happen? The only way wherein you have a duplication, but a duplication only of the exons, but not of the introns, is through retrotransposition. Therefore, the guess is that this entire thing is actually the retrotransported ADH gene, and looking at how many mutations have accumulated, etc., people have figured out that this happened about 2 million years ago. So, and this is the beauty, this entire thing has come, and also the stop codon. So, if you look at the original YMP gene, obviously there is a lot more after that, but because this stop codon has now become inserted over here, now this thing, I mean it already had a promoter in this direction, right, for the yellow emperor gene. So, this thing is now essentially behaving like a separate and independent gene, and this is what is known as the Jingwei gene. So, it comes from a Chinese myth in which a princess had metamorphosed into a new form, which is pretty much what this gene is doing. But the interesting bit is that this gene is active, it is a functional gene, and it is seen that it is expressed in the testes, and it is thought to be involved in the metabolism of hormones and pheromones, which obviously if you are a fly, these are very, very important things for your physiology. So, this is an example of exon shuffling. Now, we are going to get back and we are going to look at the third way, which is de novo gene birth. So, as the name suggests, this is not coming from existing genes, this is essentially non-coding gene sequences. You know that in the genomes of many organisms there are massive DNA sequences which do nothing. So, some of those doing nothing DNA sequences can actually lead to the formation of new genes. So, yeah, a non-coding sequence leading to a new gene. Now, how exactly will this work? In order for this to work, the new gene will need to acquire a promoter, it has to start getting transcribed, it has to have it will probably end up getting mutations, and these mutations in insertion, deletion, whatever, at some point this should create an open reading frame such that whatever is being translated, that amino acid sequence leads to something, some important or some useful product. But that is not an easy process, right. It is a pretty lots of things have to come out in the correct way, and that is why for the longest time people thought that this is not going to work. But when people started looking for it, slowly they figured out that new genes do arise in this particular way. So, when this happens, typically you are going to get genes which you are not likely to find anywhere, because obviously these are not genes which are being transmitted vertically, these are genes which have arisen de novo, and this is what can lead to species specific genes. Do we have examples? We do. So, for example, in Drosophila there are 3 genes, CG30395 which is involved in insemination, CG9284 which people have figured out is a non-coding RNA, and CG31882 which has some function in terms of sperm, birth of sperm, which basically functions in sperms, sorry. So, turns out that if you knock down any of these 3 things, then it is lethal, which straight away tells you that whatever function these genes are performing, those are essential functions. People are still trying to figure out precisely what protein and how those proteins are working, but it is clear that new genes can be formed like this. The fourth thing is of course horizontal gene transfer, where the gene is coming from something which is not within the species, from some completely different species, or sometimes an organism of the same species but not your parents. So, not a vertical transmission. Very common in prokaryotes, responsible to a great extent for all the antibiotic resistance that we are seeing around us, and it is a new crisis that is just waiting to brew. I am not going to talk about it, we have already spoken about it in the past, but what I am going to talk about is something slightly different. So, we looked at so many ways for new genes to come about. Which one is more important? Okay. Which one is less important? What are their relative frequencies? Now, obviously, it is very difficult to give one answer which will be true for all species. This true answer will most probably differ across species, it will most probably differ across taxonomic groups. However, if we can look at even one case, we will just get some rough estimate, and that is what was done by Zhu et al. In 2008, when they looked at the whole genome sequences of 6 closely related species in the Drosophila melanogaster subgroup. Okay. So, these are like Melanogaster, Simulans, Yakuba and etc. So, these are all very closely related to Melanogaster. So, that is why they are called the Melanogaster subgroup, but they are different species. Okay. So, they looked at all the new genes that are there in these species and tried to figure out what the proportions are. And when they did that, this is what they found, that duplicates about 44.1% of new genes, de novo gene origination is about 12% of all the new genes, retroposition about 10%, and chimeric gene structure through exon shuffling or whatever mechanism, that is about 30% of new genes. And they figured out that the rate of formation of new functional gene is about 5 to 11 genes per million years, which is actually very high. And if you look at copy number polymorphisms, they were able to find about 44.4% of that. So, what I am really trying to tell you is that none of these numbers are insubstantial. It is not like 0.1% or 0.01% or so. Sure, duplication is the major mechanism, but this is 12%, this is 10%, this is 30%. Okay, these are all largish numbers. So, all that this is suggesting is that barring horizontal gene transfer, all the mechanisms have actually played a major role in formation of new genes in Drosophila, and that is a very interesting piece of information. One now really needs to do similar study in other groups and see that what is the commonality. Maybe in other groups again we will see that all the mechanisms are important, or maybe some mechanisms will be more important in some groups and some other mechanisms will be more important in some other groups, who knows, right. Now, till now we have spoken about how new genes arise, and we have seen that although in some cases the rates can be pretty fast, they are still in terms of millions of years. 5 to 11 genes per 10 lakh years is about 1 per 2 lakh years or so, and that is a long time. And even Yeah, I mean, that's what I mean. Even with that, it's a long time. And although it's okay on an evolutionary scale to get a new function, this is obviously not going to work when an organism is trying to adapt to ecological or environmental changes, which obviously happens on much faster time scale. So, although it is okay for biological novelty, all these mechanisms, organisms, when they are trying to live on a day to day basis, they need things which are much faster, at least on the generational time scale faster. So, what are those mechanisms? How do they operate? And how does having those mechanisms, how do they connect with all these mechanisms for generating novelty that we saw in terms of long term evolution? That is the question that we are going to answer in our next discussion about faster time scale mechanisms. See you then.